Um, so yes, my name's Cassie. I'm a PhD student at the University of Melbourne. As Ellie said, just wrapping up my thesis now, so fingers crossed. Yeah, I'll be able to wrap it up soon. And I will be talking about my research on quantifying the topology of in vitro neural networks with graph theoretic models. So just a little bit about my lab. So I'm from the stem cell disease modeling lab. We have a psychiatry stream, so that's headed by Maria DiBiase, and also a retinal and optic nerve stream, which is headed by Elise Pive. Within psychiatry, we have a neuroinformatics group, which I'm part of, and also we've got people working on brain organoids as well. So, onto my research. So, I'm interested in morphological measures of neuron health. I work with neurons derived from human embryonic stem cells, and when we culture these cells in vitro, they like to actually organize into these networks over time. So this happens during maturation and differentiation. And we know that this morphological network structure is actually associated with functional maturity. So really its entire purpose is to kind of optimize communication throughout the network. And we know that from a lot of different kinds of studies, electrophysiological studies, calcium imaging studies, etc. Um, so the way that the morphological health of these cells is usually quantified is through what are called neurite outgrowth assays. Pretty straightforward concept. So you quantify the overall neurite length, and then supposedly the higher the length, supposedly the higher the kind of health of your neurons. But I found in my own research that this was only really valid up to a certain time point in the cell lifespan. So after the cells exit this neurite elongation phase, they tend to enter a network organization phase where you've got the somata coalescing into these clusters here, and you've got the neurites bundling into these fascicles. And because you've kind of got structures that are overlapping each other, that can actually confer a decrease in overall neurite length. And the issue is that this is actually what happens in dying cultures as well. So when you've got neurites that are retracting back into the somata, you can obviously also get a decrease in neurite length. So if we're getting similar trajectories for cells that have essentially completely opposite profiles of health, that's obviously an issue. And it led me to want to quantify instead the self-organization of these cells. So to do that, we developed a live cell imaging protocol to track neuron self-organization. So as I said before, I was working with human neurons. They're actually a reporter line, so they were just tagged with GFP. And we find when we're imaging a reporter line over a long period of time with fluorescence imaging, that there tends to be a lot of cell death due to something called phototoxicity, which is where the cells and, like, to a certain extent, the media, they produce reactive oxygen species, and that can actually really affect the viability of the culture to a pretty substantial extent. So because of phototoxicity, a lot of well, essentially all of the live cell imaging protocols for neurons, they're usually capped out at about 2 weeks. So with this protocol here, I was actually able to get the cells to survive for 30 days, so just over 4 weeks. And all I did was I optimized 3 factors in the culture microenvironment. So the substrate that the cells were grown on, so mouse versus human laminin. The media, I was comparing this conventional neurobasal media to this new brain fizz imaging media, and also the seeding density, so just 100,000 versus 200,000 cells per centimeter squared. Both of those are actually high densities, but I kind of needed that density to get kind of optimal network organization in the culture. So just looking down here, so if I've just got two time points, so day 19, day 29. The best conditions where the cells tended to kind of survive for the full 30-day period were the ones that had mouse laminin or brain-fizz imaging media, or the combination of the two. And you can see with those, there was kind of extensive self-organization. And I found this really interesting negative interaction between human laminin and neurobasal media. So you can see that by day 29, the cells were essentially all dead, so that we've got extensive kind of blebbing and neurite retraction. And if we look at the neurite length measures and we isolate for those two conditions, you can actually see that the trajectories are quite similar to what we see for all of the other conditions. So in one, we've got dying cells, one we've got self-organizing cells, and yeah, they essentially look the same. So this is just essentially corroborating what I was talking about before. So as I said, that led me to want to quantify the actual degree of self-organization of these cells. And I did that by quantifying the network morphology. I did that in two ways. So first, just general morphological network indicators like neurite fasciculation and somata clustering, and also through building microconnectomes. So these are network graphs that are generated by mapping the somata to nodes and the neurites to edges. And this is now a formal mathematical object, so you can actually study the topology of this through graph theory. And I'll just touch really briefly on the morphological network indicators. I've just got one slide, and then I'll focus most of the talk on microconnectome building. So I derived a range of indicators. These are the two that kind of pertain most closely to the network organization. So the first was the cluster density factor. So this was an algorithm that I designed to compare the clustering behavior at time point n to the first time point. So it's essentially kind of estimating the density within these clusters here compared to the somata area at the first time point. And we would expect a higher CDF for cells that are more self-organized. And then also there's this neurite coherency measure, and this workflow actually uses an algorithm by Pusprokey. And just in this image here, I've got the neurites colored by orientation. So essentially what coherency quantifies is the amount that the neurites kind of organize in the same direction. So up here, there's kind of two scenarios in this image. Up here, we've got the like a more kind of disorganized morphology where we've got a wider range of angular orientations present. And then down here, we've got this fasciculation. So we've got a relatively constant angular orientation along the entire length of the fascicle. So this kind of section down here would have a higher neurite coherency. And I use these two indicators to quantify the phototoxicity conditions that I was talking about before, so laminin media seating density. And I've just ranked each of the condition by these network indicators. So you can see that the three top ranked conditions all had this brain-fizz imaging media in them. In a separate viability assay that I conducted kind of towards the end of the culture, brain-fizz imaging media actually came out as significantly higher than neurobasal, and it was the only factor that was significant out of laminin and seating density. So there's a really high alignment between the viability assay and these network indicators here. So yeah, that's good. That's kind of reaffirms that it's telling us something meaningful about the health of the culture. So onto building the microconnectomes. So kind of when we're just working with 2D images, you can't obviously penetrate the Z depth of thicker structures. So we couldn't actually penetrate the Z depth of the clusters in the network, and they could get quite large. So the kind of largest ones I had were up to 500 or 600 microns per cluster in kind of diameter. And the way that we worked around this was to generate a hierarchical network reconstruction where we take this mesoscale image and we subsample these clusters with 3D super resolution microscopy to just get an idea of their kind of internal structure. So this is just a really high-level overview of the network reconstruction pipeline for both 2D and 3D images. I did some pre-processing in Fiji, segmentation with Elastic, which is this really great semi-supervised AI tool. Did a really stellar job of segmenting images even with very poor signal. So I was very glad to be using that. And then we mapped the somata to nodes and the neurites to edges to get this overall hierarchical network reconstruction. So we did come across a couple of challenges in this pipeline. The first being the neurite crossover problem. So this is actually quite a well-characterized issue in neuroinformatics, and it occurs mainly in 2D images, some 3D images, but mostly 2D. So it describes scenarios where you've got neurites extending from different somata that each come to this kind of crossover point, or this juncture point in the middle. We as humans can very easily kind of distinguish which one of these neurites belongs to which one of these cells, but it can be kind of difficult for, say, for example, a network reconstruction algorithm or a segmentation algorithm even to make that call. So I've just got a little example down here. So if we take network reconstruction, for example, most of the algorithms are just based on connected components. So if a path exists between the structures that you're denoting as nodes, that means that an edge will be formed there. So if we take, for example, this somata and this somata, technically there is a path between them. So technically an edge would be formed between those two, which we can see is incorrect. We would want our technique to be able to individuate the neurite structures in this way to get this particular edge configuration. And yeah, as I said, really well-characterized issue. This is not my work. This is a review by Hung, just looking at algorithms to solve the neurite crossover problem. One thing that they all have in common is that they all attempt to solve the problem at each individual crossover point. So if they kind of all iterate through the image, identify each point, and then attempt to solve it, usually through some combination of geometric principles. But yeah, this means that they're usually quite computationally expensive, and they're not really suited to high volume or high throughput data sets. So I wanted to design an algorithm that made this process more efficient. So I took more of a global approach. So if we take the overall neurite mask, I separated out the mask into orientation-specific masks. So each one of these structures falls within a specific angular window. And then the idea was to map each one of these masks to a specific network. So I had kind of orientation-specific networks that I then concatenated into an overall network structure. And the principle there was to essentially that if you have neurites that are all like kind of on the same filament, then they would have a relatively consistent angular orientation. So they would fall within the same angular window. The algorithm then kind of closes this gap created by the juncture point by just a linear structuring element. So it's just positioned at the same angular orientation as the segment. And then this produces a continuous neurite filament that I then was then able to assign the edge between these nodes just using a normal connected components approach. And with this technique, we were able to process each crossover point in 1 to 10 milliseconds. So that means we could process 10,000 crossovers in approximately 100 seconds. And I also just wanted to touch on the process of neurosphere integration as well. So if we take our wide field image, the pipeline iterates through each one of these somata structures and classifies them into either a single somata or a cluster. And then if a cluster is detected, we kind of initiate this embedding process. So the first step is to select a size equivalent neurosphere. And just to give you an idea of the library that I acquired for our specific project. So it was a library of 75 neurospheres, over 20 terabytes. I think it was 21 terabytes. And I was also looking at 2 disease models. So I'll talk about those in a second, but they were also included in this library. And you can really see each cluster kind of has its own personality. So it was really important to try and capture this variability to make the library as representative as possible. So moving on to the second step, we wanted to generate a 3D network from this neurosphere that's been selected. So to do that, first to map the neurites to edges, I was able to just use a normal, just a pretty standard Otsu thresholding approach to get these kind of segmentations. I'd been working with live cell imaging data before this immunostaining data, so the live cell imaging data was very patchy in some places, so it was really nice to be working with these markers that had like a really nice robust signal across the entire neurite structure. Nuclei were a different story. So they, in the neurospheres, they tended to pack up really tightly against each other, and there was a lot of kind of contact between the nuclei. So if I just use a normal segmentation approach, I would essentially get this giant connected component, which was not of any use to me. So instead, what I did was I used StarDist, which is a deep learning based Python package to individuate each one of these nuclei. And that served as the basis for the 3D network reconstruction. And then next step was to just flatten this 3D network to 2D, calculate a convex hull. And then this is kind of the embedding process. So the idea was to position the super resolution convex hull, which is in blue, over the cluster site in the wide field image, which is in green here. And that was actually done at a random spatial offset. So that was to include some of that kind of biologically realistic variability that I was talking about before. I've just got 3 iterations of that here. And then the idea was to clip the nodes that fell outside of this cluster site to get a network that exactly matched its size. And then it was just embedded into the mesoscale network to generate this overall hierarchical network. So I wanted to see whether I could use this network reconstruction method for disease phenotyping. So I generated 2 disease models, an epilepsy model and a schizophrenia model. And the idea with kind of pharmacological disease modeling is your target drug specifically perturbs molecular pathways that are implicated in the pathology of the disease. So that's kind of the principle there. For the epilepsy model, I used caenic acid, which is a glutamatergic agonist, and that induces pretty widespread activity in the culture, kind of like a seizure-like event. And also I generated a schizophrenia model with MK801, which is a glutamatergic antagonist, and that just induces neurite degeneration. So what we found in the network analysis is that really distinct patterns actually emerged across the groups. I've just got 2 time points here, so day 7, day 25, and within each time point, I've got the somata neurite segmentation, the overall community network structure, which I'll talk about in a sec, adjacency matrix, and the co-classification matrix, which relates to the community structure. So essentially, these two metrics are quantifying the grouping behavior of nodes into communities. So the idea is that the connectivity within communities is always higher than the connectivity between them. And this can be calculated across different granularities or resolutions. So your kind of traditional detection algorithm, like the Newman algorithm, that just detects it at a fixed resolution. So that's this gamma parameter down here. But you can also get algorithms like this one from Dube that detects the communities across a range of granularities. So you can see from coarse to fine granularity. And then what it does is it calculates the stability of the community identity of each one of these nodes across resolutions. So that's what's being represented in this co-classification matrix here. Sorry, just going back. Just in the control cultures, you can see, as we expect, extensive self-organization. The co-classification matrix, these really kind of like robust, stable blocks emerged in the matrix, and that's just indicative of these highly stereotyped cluster structures in the culture. And if we compare that to the matrix at a younger time point when the cells are more disorganized, you can see that the matrix is a bit more kind of irregular and unstable. So that's just essentially telling us that the nodes are essentially swapping communities as the resolutions increase. The schizophrenia model was very different. So we got minimal self-organization by day 25. I think that manifests really clearly in the amount of communities that were detected. So you can see that the number of communities doesn't really change from day 25 from day 7. And if we compare that to the healthy controls, you can see that there's a massive decrease from day 7 if we compare it here. So yeah, that's essentially capturing the fact that the cells all coalescing into increasingly larger and larger clusters in the culture. And then in alignment with the consistent disorganization in the schizophrenia model, we have quite an irregular co-classification matrix across both time points. The epilepsy model was kind of in the middle of these two models. So we got self-organization, we got these robust blocks emerging in the co-classification matrix. One key difference, though, was the sparsity of edges within the communities was a lot greater. So if you look at, for example, like this red community here, you can see that the edge density was actually much lower than for example, like this orange community within the controls. So yeah, that was one key feature that emerged. And I wanted to quantify graph theoretical metrics over the lifespan of the culture as well. So here we've just got the metric over the observation period where I've just fitted a polynomial curve. Down here we've got the area under curve for each one of these diseases. And I've also, just kind of in terms of the metrics that I looked at the overall edge density, the intra-community edge density. So you can actually see those are quite similar. And that's just because the community organization really drove a lot of the overall network organization, number of communities, and also the mean edge distance. So in healthy controls, we found that there was a low number of communities, especially in more mature stages, which we would expect, and also a high intra-community edge density, as you can see here, and also a high overall edge density as well. The schizophrenia models were essentially the exact opposite of this. So quite a high number of communities and a low overall network edge density and low intra-community edge density. And a really interesting feature emerged in the epilepsy models. So we found a high mean Euclidean edge distance. So you can see that here. And that actually fits in with a theory that as the cells are kind of undergoing these seizure-like events, they're actually sprouting new neurites consistently across the lifespan of the culture. And because they are being so consistent in sprouting them, it means that they don't actually have time to self-organize. So this is kind of in contrast to the healthy controls where you've got that cluster, and within the cluster, you've got kind of like an overabundance of short range connections, and that really drags down the mean edge distance. So yeah, that was likely driving the higher edge distance that we saw in the epilepsy models. And I also characterize the small world characteristics of these networks, which essentially is just looking at the efficiency of them. So there's two kind of main small world indices that people look at. The first is the characteristic path length, which is if you kind of visualize a signal propagating throughout the network, it's the amount of steps that the signal has to take to reach its destination. So generally, lower values are more efficient. And also the clustering coefficient. So that's the amount of closed triangles in the network. So the way that we can determine whether our empirical neuron networks show small world indices is through comparing them to synthetic networks with known small world characteristics. So I've just got two networks that I've generated here with the Fruchterman and Riemann algorithm. So these are synthetic versions of this empirical neuron network. So they show they have like matched edge and node densities. And in a random network, we know that the characteristic path length will always be low. And that's because it's got quite a number of long-range connections. So that means that the signal will usually have a shortcut to choose as it's propagating. But it also has generally a low clustering coefficient. So that just kind of speaks to the low kind of local efficiency of these random networks. The lattice networks are biased towards short-range connections, so clustering coefficient would be very high. But the characteristic path length is also high because we don't have those shortcuts that I was talking about. So if our empirical neuron network is small-world, we would expect it to have a low characteristic path length and a high clustering coefficient. So I have just quantified these small world indices for the first 3 time points in the culture, so day 7, 10, and 13. And that's because when you kind of get networks that have an edge density of over 40 or 50%, the small world indices can actually lose their sensitivity. So we were actually kind of finding that in the mature cultures. So across these first 3 time points, the characteristic path length for the empirical network was equitable to the random network, as we would expect. And the clustering coefficient was equitable to lattice networks, which we would expect as well. So yeah, this was actually the first time that a small world phenotype has been detected in neurons as they're differentiating from stem cells. And it's kind of significant because in actual brains in vivo, they typically have small world topologies. So this might be giving us an insight into what's happening in neurodevelopment in the fetus. And I just really quickly wanted to touch on the data infrastructure that I used. So, as I said before, working with 20 terabyte data sets, even my lifestyle imaging data sets were usually over 100,000 images. So it's very, very important that I streamline this infrastructure as much as possible. So I'll just go through it. So I was using AriScan imaging for my super res data set. So I had to do AriScan processing and tile stitching. And I did both of those on the Windows workstations at BOMP. All of my data was stored on a MediaFlux server. And then I used a HPC platform for most of my processing. The platform at Unimelb is called Spartan. It's a really great system, very flexible. So I was just using mostly the physical nodes, but I know that there are virtual nodes also available. And the very kind developers at Unimelb have blessed us with these command line interface clients that really rapidly transfer data. So it's done across kind of multiple threads. And I was using that extensively throughout my project. It was very, very helpful. So generally to run this pipeline from start to finish, excluding the neurosphere analysis, would take me about 2 weeks. I was running it across generally 8 cores per disease model, so actually it's 24 cores in total. So yeah, 2 weeks, so nothing too scary. So yeah, just to summarize, I developed a live cell imaging protocol to track neuron self-organization over time, and I did that by reducing phototoxicity in the network. We developed some tools to quantify just overall morphological network indicators, and if you're interested, most of these tools are on my GitHub page here. So those tools encompassed quantifying the amount of neurite fasciculation on top of the degree of somata clustering. And we also generated a hierarchical network reconstruction pipeline that synthesized dual resolution microscopy data sets. And we were able to use that pipeline to identify key network phenotypes in schizophrenia and epilepsy models, and to also identify emerging small world topologies in neurons that are differentiating from stem cells. And yeah, I'd just like to really thank everyone that's been involved in this project. First of all, Ellie, as she said, she was one of my supervisors, and I really learned a lot from her about this project. So thank you, Ellie. Also, Kalyan and Paul from BOMP. And my other supervisors, Maria, Andrew, and Kayo. And also just a particular shout out to Simon Maxauer from the University of Wollongong, who helped us really extensively at the start with our neuron differentiation protocols. So thank you, Simon, wherever you are. And yeah, happy to take any questions.